I'm a biochemist, actually, a metabolic biochemist. But many years ago, I became interested in fatty acid nutrition and fatty acid biochemistry. And I've been running a research group in the area of fatty acid handling, fatty acid metabolism, fatty acid functional effects for probably 25 years or so now. So I'm based at the University of Southampton in the Faculty of Medicine. We have, just out of interest, a NIH Biomedical Research Centre in Nutrition in Southampton. It's the only one of those in the UK. So I guess you would say I have some expertise in omega-3 fatty acids. I was asked to talk about these fatty acids in the context of inflammation, which is actually one of the areas that we do a lot of research in. So that's what I'll do. If you are academically minded and you want to find out a little bit more detail, pretty much everything I'm going to say in my presentation is in these two publications. This is one in the British Journal of Clinical Pharmacology from 2013, volume 75, number 3. And the other one actually is just available online at the moment. It's in a journal called Biophysica Acta. So this is a really up-to-date review on omega-3 fatty acids and inflammation. If you do a search on me in PubMed, this will be quite near the top of the list because it's only come online relatively recently. Okay, so inflammation is a normal process. It's part of the way that people deal with insults. There's an inflammatory response which can be measured in many ways, and some of you may have some experience in that. We'll talk about some of those things shortly. This response is initiated by the presence of a trigger. Most often we think of this as what I've called here microbial components. That is something to do with an infection. So this is part of the response, the immune response to infection. The inflammatory response can also be triggered by tissue damage, physical tissue damage, metabolic tissue damage, and also by metabolic stress. And what's interesting is even if you give somebody a high-fat meal, you can measure a rise in inflammatory markers in their bloodstream in the few hours after consuming the meal. And actually we're doing a lot of studies using that model to probe inflammation in normal weight and obese people and so on. The link between the presence of these triggers and the response is, of course, the molecular and cellular events that are initiated by the presence of the trigger. So there are, on the surface of many types of cells, receptors that respond to the presence of these triggers. They initiate what we call cell signaling processes, so things happening in cells, and maybe I'll talk about some of those things as we go along. And eventually those events change the pattern of genes that the cell receiving the stimulus is expressing. So then the cells start producing a different profile of proteins. And those proteins can appear in the bloodstream, and you can measure those as a readout of the response. So there's lots known, of course, about these molecular and cellular events. And as I say, I'll probably touch on some of these as we go along, but they're not going to be really a central focus of what I'm talking about. Now, if you look in a little bit more detail at what's going on with cells in the context of an inflammatory response. So going through the middle here, this is the bloodstream. So this is a bit like, a pipe running across the slide here. And the blood vessels along their walls have these endothelial cells, so they're separating the blood from the underlying tissue. So here I have some different tissues on either side of the blood vessel. The white blood cells, or leukocytes, can interact with the endothelial cells, and this interaction is enhanced if there's inflammation around. And what happens is these white cells, which are moving through the bloodstream, as you might imagine, in the flow of the blood, actually can bind to the endothelial cells, and then they cause little gaps to appear between endothelial cells, and they squeeze through those gaps, and then they're into the tissue or the vicinity of the tissue. And this is where they might be exposed to the trigger that I've been talking about. It could be an infection, it could be something else. And they become activated. And when they're activated, they start releasing sometimes very quickly and sometimes with a time delay. Many, probably hundreds, maybe even thousands of different chemicals. And those chemicals have many roles in the inflammatory process. Some of them actually serve to influence the endothelium to make this process more efficient or more effective. In other words, to bring more troops in the army into the site of inflammation. So there's all sorts of things happening there. Some of them actually are damaging to bacteria, so that's actually why they're made to kill the bacteria that are there, for example. Some of them reorganize the tissue, so they change the phenotype of the tissue and so on. Now, all of this has got to be controlled, and if it's not controlled, they actually cause damage to the local tissue. So these are chemicals that are damaging, and they can damage not only invaders like bacteria, but they can damage the host tissue as well. Some of these chemicals spill over from the site into the bloodstream, and you can measure increased concentrations of these chemicals in the bloodstream. So, for example, let's say this is the joint of somebody with rheumatoid arthritis. All of the inflammatory activity is going on in the joint, but you can actually measure high concentrations of some of these chemicals in the bloodstream because they've spilt over from the joint into the bloodstream. Same with inflammatory bowel disease. Same with asthma. Even though the inflammation is in the lungs, you can measure higher concentrations of these markers in the blood. So that's handy for tracking inflammation because it's easier to sample blood than these different tissues. Okay? The important thing about this, not just to make it convenient for people like me to measure things, is that once these chemicals are in the bloodstream, they can actually affect other tissues away from where the inflammation is. So some of you may know that people with inflammatory bowel disease suffer bone loss. And one of the reasons they suffer bone loss is that the inflammatory chemicals go from the bloodstream to bone and start increasing the rate of bone turnover. The other thing that you will probably be well aware of is these proteins called acute phase proteins, like C-reactive protein. These are released from the liver, and these are really good markers that inflammation is going on somewhere in the body. So again, you get the chemicals spilling over from the site of inflammation into the bloodstream. In the case I'm talking about, they go to the liver and they change the types of protein that the liver is making, and you get an increase in acute phase, or a change in acute phase protein concentrations in the bloodstream. Things like C-reactive protein, fibrinogen, and so on. The 5 cardinal signs of inflammation are heat, redness, swelling, pain, and loss of function. These were recognized over 2,000 years ago by the clever Greek physicians. All of these signs of inflammation are caused by the chemicals that are released as part of the inflammatory response. So the swelling in the joints of someone with inflammation, the pain of a headache, all of those things are caused by those chemicals released from those cells. I started off by saying inflammation is normal. It's part of what we need to deal with the presence of pathogens and so on. But it's quite clear that when inflammation is uncontrolled or excessive, or it continues without control, tissue damage and changes in metabolism occur. So these could be local tissue damage, like to the joints of someone with inflammation, the gut mucosa of someone with IBD, the lungs of someone with asthma or COPD. They could be distant. I talked about the bones. And also there are changes in metabolism. So eventually actually you get loss of skeletal muscle mass and body wasting. And that's a feature of the inflammatory drive of cancer bearing, for example. So cancer cachexia has a strong inflammatory drive. So as a result of all of this, it's now recognized that there are lots of diseases and lots of conditions that involve inflammation. And I mentioned some of these already. They obviously include classic inflammatory diseases like arthritis, inflammatory bowel disease, psoriasis, and so on. And of course, these diseases are treated with varying effectiveness with anti-inflammatory medications. So that's a direct attempt to control the inflammation because the inflammation is involved intimately in the pathology. So we see all these things here: asthma, allergic diseases, COPD, and so on. But on this side are other diseases which sometimes people find surprising that inflammation has a role. So probably about 20 years ago, it was recognized that inflammation is central to the growth of the atherosclerotic plaque in the blood vessel wall. It's not the only thing involved, of course. There's lots of other factors involved, but it's one of the key things. Acute cardiovascular events like having a heart attack can involve inflammation. Response to surgery, injury, trauma, neurodegenerative diseases, so it's now recognized that Alzheimer's disease and other neurodegenerative diseases have an element of inflammation. Some cancers, body wasting, I mentioned cachexia, non-alcoholic fatty liver disease. Obesity is now recognized to involve inflammation, although whether that's causal or whether that's an effect of having too much adipose tissue is not really clear. People are arguing that out. So this is the front page of Scientific American. Actually, it's from 2002, so it's a little bit old now. But on the front cover, a fire within, inflammations linked to heart attacks. So this is the key feature in Scientific American of about 12 years ago. This is Time magazine. I think it's also about the same time. The secret killer, the surprising link between inflammation and heart attacks, cancer, Alzheimer's, and other diseases. I guess you're all interested in omega-3 fatty acids, or you wouldn't be here. The omega-3 fatty acids that I'm going to concentrate on are the fishy omega-3 fatty acids, eicosapentaenoic acid, EPA, and docosahexaenoic acid, DHA. These are unique fatty acids in terms of their structure, because they have, if you remember your lectures on fatty acids that you had, for some of you not very long ago, looking at you, these have very long chains, and they can have unsaturated bonds within the chains. And these omega-3 fatty acids are 20 or 22 carbons long, which is quite long, and they have many double bonds in the chain, either 5 or 6. The term omega-3 refers to the position of one of the double bonds in the chain. So that's a structural feature. It's not a magic functional feature, although a lot of people might wish that it was. It's a structural feature. So it's just boring biochemical description, omega-3. They're also really quite unique in their dietary sources, because the only naturally rich source of these fatty acids is, for most people, is fatty fish. Okay, they're also found in seal meat, they're also found in whale meat, but not too many people outside of 1 or 2 countries like Norway and Iceland eating those sorts of things. So they're unique structurally, and they're unique from a dietary point of view. Okay, obviously they're also found in supplements that are derived from fatty fish, and a few other things as well. So the working model that we have in my lab is that if we increase what I've called the supply, in other words, give people more of these things, or indeed give them less, but change the amount that they are administered, we can do something about the structure of the cells that are involved in inflammation, and I think I'll tell you something about that shortly. We've been studying that a lot over the years. Trying to find out about the features of the time course and the dose responsiveness of incorporation into inflammatory cell membranes, and the functional effect of that. So you'll remember that all cells have this lipid bilayer around them, and that lipid bilayer is composed mainly of phospholipids, and phospholipids have 2 fatty acids linked to them. So I'm talking about getting omega-3 fatty acids into those phospholipids in the cell membrane. The reason that's important is the type of fatty acids linked to the phospholipids actually affects the structural and functional aspects of what the cell membrane is doing. So it actually affects the physical nature of the membrane. Some of you may have heard of this term called membrane fluidity, how liquid a membrane is. Scientists don't like this word fluidity, they like the word order, so how ordered the membrane is. So if you think about sources of fat, and you have some butter, and you have some soybean oil or rapeseed oil, okay, they are both almost, well, I don't know what they are, but 90% plus of fat. But one of them is solid at room temperature and one of them is liquid at room temperature. And that physical feature is determined by the fatty acids that those things are made of. So in the vegetable oil, there's lots of polyunsaturated fatty acids. They're liquid at room temperature. In the butter, there's lots of saturated and monounsaturated fatty acids. They're solid at room temperature. So if you think about a cell membrane, is somewhere in between those two things. And by changing the fatty acid mix in the membrane, we can drive the membrane to be more or less liquid, if you like, more or less ordered. So you can change the physical nature of the membrane. That and other features actually affects how the membrane behaves when a cell is stimulated. And this is very important in the context of inflammation. So membrane behavior. Then you have these pathways that I talked about before, these cell signaling pathways by which events are happening sequentially within the cell in response to the stimulus, ultimately hitting the nucleus and changing the pattern of genes that are expressed. So this is also influenced by things happening in the membrane. And then finally, I'm going to say something about chemicals produced from fatty acids. You're all familiar with these chemicals. And the type of fatty acids present in the cell membrane influences the production of these chemicals. These are chemicals like prostaglandins, leukotrienes, and so on. So anyone who uses aspirin, of course, is directly targeting the production of prostaglandins, because aspirin works by inhibiting the production of prostaglandins from the omega-6 fatty acid, arachidonic acid. So aspirin is an anti-inflammatory drug that directly targets omega-6 fatty acid metabolism. So I'll probably say a little bit about that. The reason I say probably is I can't actually remember what's in my talk until I see the next slide. So we'll see how I get on. Don't worry, that's not especially for you. I'm always like that. So you're changing things from the membrane to the nucleus, the chemicals that the cell is making. Therefore, you're changing the way that the cell is responding to these triggers. You can make it respond more vigorously or less vigorously, depending on what you do. That's our thinking here. And therefore, you can change the response as a whole. So you could have an impact on some of these conditions here. And hopefully that's where I'm heading. So if you go to the lab and you measure the sorts of fatty acids in the white cells taken from the blood of people like yourselves, then this is the sort of picture you get. So what we do is we express the different fatty acids as a proportion of the total. So altogether there's 100% of fatty acids in the membrane of these cells. That's the way we express it to make it simple. So these are the omega-6 fatty acids. Okay? So this is proportion of total. So you see there's an awful lot of this fatty acid here. That's arachidonic acid. 20 carbons and 4 double bonds, omega-6. 1 in 5 of the fatty acids in your inflammatory cell membranes is arachidonic acid. That's an awful lot of one fatty acid. You don't eat that much arachidonic acid by and large, but these cells concentrate it in their membranes. There are some other omega-6 fatty acids, linoleic acid, the common vegetable omega-6 fatty acid found in margarines and in most vegetable oils. So altogether we're talking about 30-odd% of these fatty acids. So you've got plenty of this arachidonic acid in your cell membranes, around about 1 in 5 of the fatty acids present. When the cell is, that's here, when the cell is stimulated, that arachidonic acid is released, and then it acts within the cell as a substrate for some enzymes called cyclooxygenases, lipoxygenases, and cytochrome P450s. And these enzymes produce a whole family of chemicals called prostaglandins, thromboxanes, leukotrienes, and so on. Some of you may have heard of these chemicals. So leukotrienes, for example, are one of the key mediators involved in asthma. And some of the drugs that people are trying to use to treat asthma actually block the production of leukotrienes or block leukotriene receptors. I already mentioned that aspirin works by inhibiting cyclooxygenase activity. So anything that you would take aspirin for involves these chemicals here. Okay? So immediately you see there's a link here between this omega-6 fatty acid and inflammation. Because I know that these things are involved in inflammation, because I know that there are drugs that people are commonly using to control the production of these things, and the things they're trying to control are something to do with inflammation. So there's a direct link here. Incidentally, these chemicals are all in a family called eicosanoids. Okay. Now if we look at the omega-3 fatty acid content of our cells, this is the sort of picture we get. So there's not very much of the common plant omega-3 fatty acid, alpha-linolenic acid. Usually you can hardly measure that in cell membranes. EPA, eicosapentaenoic acid, let's say about 0.5%. DHA, 2 or 3%. So you can measure these in pretty much everybody, but the amount is variable, and the amount is typically much lower than the amount of the omega-6 fatty acids. So in other words, most people have a much higher content of omega-6 fatty acids in their cell membranes than they do omega-3 fatty acids. And of course, most people have a much higher content of omega-6 fatty acids in their diet than they do of omega-3 fatty acids. Okay? Probably actually almost everybody does, in fact, because omega-6s are very prevalent, and omega-3s, as I said, are mainly found in fatty fish. Now, we can change this by giving people omega-3 fatty acids preformed. And you could do this actually by feeding people salmon every day, if you wanted to do that. People have done experiments like that. Or you could give them omega-3 supplements, which is easier to do in a study because you can actually do a randomized controlled trial of an omega-3 supplement compared with olive oil or corn oil or palm oil or whatever. So I'm going to show you some data of those sorts of studies. So this is a study where we gave healthy people omega-3 fatty acids, 2 grams of EPA and 1 gram of DHA per day, every day for 3 months. And this is the amount of arachidonic acid, that inflammatory chemical precursor, and of EPA in the membranes of their white blood cells before the supplementation in red and after the supplementation in yellow. So you'll see there is a small, but it's a significant reduction in arachidonic acid, and there's a large rise in EPA and also in DHA, which isn't shown here, and that changes the ratio of arachidonic acid to EPA quite markedly. And probably this sort of ratio is quite important in determining what the cells are doing. Because these are two, if you like, alternative fatty acids that end up having different functions. So the relative amounts of those two are probably quite important, which is something hopefully I will draw out of my talk. So this is what happens after 3 months. So people are often interested, well, how long does this take? So this is the time course of that change from that study. So this is the starting point after 4 weeks, 8 weeks, and 12 weeks. This is for eicosapentaenoic acid, EPA, and docosahexaenoic acid, DHA. Then we stopped the supplementation at this point here, and we took another blood sample 8 weeks later. So what you see for both EPA and DHA is they get into these cells within 1 month. Okay, so you've got as much into the cells as you're going to get after a month. Okay, now this timing is actually a feature of the cell type. So cells that turn over very quickly incorporate these fatty acids very quickly, and cells that turn over slowly incorporate them more slowly. So if I was to show you the same data for red blood cells, which turn over more slowly than white blood cells, this line would be coming up here and probably would not yet have peaked. Okay? If I was to show you this for platelets, which are turning over very quickly, although I don't have the earlier time points, it would have come up here and peaked very early. Okay? So anyway, in this study, after a month, both EPA and DHA had reached a new peak. Okay? And they stayed like that, continuing to take the supplement, didn't change things very dramatically. Then we stopped, and 8 weeks later, went back, and EPA had returned to where it started. Okay? So EPA gets in when people increase their intake, and it comes back out again when they stop their intake. Okay? So you can't, if you want to think of it simply, you can't eat salmon regularly for 6 weeks and then stop and expect to keep getting the benefit. You can't take an omega-3 supplement every day for 8 weeks and then stop and think I'm still going to get the benefit. Okay. DHA is just a little bit different, and this is a subtlety that people like me are very interested in. So it gets in just the same, but actually when you stop the supplementation, the cells hang on to the DHA a little bit more than they hang on to the EPA. Eventually, this actually does come back down again. But people have interpreted this to mean that DHA is much more important to these cells than EPA, and they're trying to hang on to it. And there might be something in that. So you can increase the amounts of these fatty acids. Remember, well, I didn't say this, but it makes sense. The cell membrane is a fixed size or a fixed volume. You can't make a membrane bigger or smaller by putting more lipid into it, as a generalization. So when these fatty acids, EPA and DHA, are going into the membrane, something must be coming out. In other words, they must be replacing something. And this is a study we did. This is looking at neutrophils, which are another type of white blood cell. Again, with a dose-response study design, showing this nice dose response of EPA and DHA. So in other words, the amount that gets into the cells depends upon the amount that you're consuming. So if you give people 1 gram per day, you get a certain increment. If you give them 2 grams a day, roughly speaking, you get twice that increment. If you give them 4 times a day, you increase that increment another twice, so 4 times the normal. Okay? So there's a dose-response relationship. In other words, the more people eat, the more they end up within their cells. And this is true for platelets, for red cells, for white cells, for heart tissue, and what have you. People have looked in many different places. And characteristically, as these fatty acids get in, they replace arachidonic acid. And remember, arachidonic acid is the precursor for these highly inflammatory chemicals like prostaglandins and leukotrienes. So that's pretty interesting. So the upshot of all of that is a functional effect. The cells behave differently. So this is a study where we fed mice diets containing different fatty acids, including fish oil here, omega-3 fatty acids. Then we took inflammatory cells from those mice. We put them in a test tube and we exposed them to a classic stimulus that's going to make them make a lot of inflammatory chemicals in that test tube. Okay, it's like an inflammatory explosion. Okay, so the readout here is prostaglandin E2, the cyclooxygenase metabolite that is inhibited by the presence of aspirin. Okay, so essentially, it didn't make any difference what we fed these animals unless we fed them omega-3 fatty acids. When we fed them omega-3 fatty acids, these cells made much reduced amounts of prostaglandin E2. Okay? So it's a little bit like aspirin, but of course aspirin works very quickly because it's inhibiting an enzyme. Omega-3 fatty acids don't work quickly because they don't work mainly by inhibiting the enzyme. They work by this removing the substrate over time. Okay? Actually, if we put aspirin in these reactions, this would come way down here somewhere. But anyway, this is pretty nice. This is mice, of course. You might say, well, show us some real animals. So this is a study in humans. This is a dose-finding study with a very eicosapentaenoic acid-rich material. We gave people placebo, 1.5, 2.5, or 4 grams of EPA per day for 3 months. We then took the white blood cells, we put them in a test tube, and we exposed them to the same agent that elicits this inflammatory explosion that I talked about before. Red is before supplementation, and yellow is after supplementation. So a couple of things. In the placebo group, there's no change, which is what you want to see. At this dose, which is about 1.35 grams per day, actually there's no effect. But at 2.7 grams per day, there's a significant reduction. It's about 30%. And there's about a probably 60% reduction at the higher dose. So there is a dose-response effect. But it looks like, at least in this particular study, you have to get beyond a particular intake before you have a functional effect. So maybe the fatty acid composition change induced by this dose is not enough to have an effect. But the fatty acid composition change induced by this dose is enough to have an effect. So there's a sort of threshold beyond which you have to get. Now, I wouldn't want to generalize this threshold for every effect of omega-3 fatty acids. Okay, this is just about, probably shouldn't even generalize it to inflammation, but this is just about an inflammatory readout. If you relate the amount of arachidonic acid in these cells to their ability to produce prostaglandin E2, there's a very close relationship. If you relate the amount of EPA in the cells, there's a very close inverse relationship. So one way that omega-3 fatty acids work to modify inflammation is they target the main precursor of inflammatory lipid chemicals, that is arachidonic acid. They reduce the amount that's present actually in the membranes, and actually they do have an effect on these enzymes, but the main effect is reducing the substrate availability. This has actually been known for a very long period of time, actually since the early 1980s. So these fatty acids exert an anti-inflammatory effect by inhibiting the production of these potent eicosanoid chemicals made from arachidonic acid. Now it turns out, because EPA has a similar structure to arachidonic acid, it's also a substrate for cyclooxygenase and lipoxygenase enzymes. So it also gives rise to prostaglandins and leukotrienes. Okay? These have a different structure than the ones produced from arachidonic acid because the substrate fatty acid has a different structure. This structural difference has a very major impact on the biological potency of these chemicals. So I've tried to summarize this here. So these chemicals made from arachidonic acid are very biologically active. They're strong. They're powerful. The ones made from eicosapentaenoic acid are weak. Okay? Some people will say they're anti-inflammatory, but actually, by and large, these chemicals are pro-inflammatory, but very weakly so. So what you're doing is you're really changing from a soup of powerful inflammatory mediators to a soup of reduced inflammatory tone. Now, this is an old study, just to demonstrate for you. So I always like to give people the evidence. What I just told you. So this is a comparison between leukotriene B4, which is a chemical made from arachidonic acid, and leukotriene B5, which is a chemical made from eicosapentaenoic acid. This is an experiment in a laboratory, and the readout here is the movement of white cells towards the chemical. So leukotriene B4 is a very potent chemical at attracting inflammatory cells to sites of inflammation. Remember, I talked about that very early on, the attraction of the white cell to the place where inflammation is happening. And leukotriene B4, made from arachidonic acid, is one of the main chemicals that does that. So this is increasing movement as a function of increasing concentration. So leukotriene B4 is, let's say, is good at doing that. Leukotriene B5 is much, much less potent. So you need maybe 100 times as much to elicit the same effect. So typically, the chemicals made from eicosapentaenoic acid are weak in their action compared to those made from arachidonic acid. This has been studied in a lot of detail. I won't go into this, but this is a paper from some American researchers from 2007 where they studied this potency of the chemicals made from arachidonic acid compared with EPA. And they demonstrated quite clearly that no matter what aspect they looked at, the chemical made from EPA was always, or almost always, much less potent than the one made from arachidonic acid. So far, I've been talking about things that kick inflammation off, the initiation phase, attracting cells to sites of inflammatory activity, activating cells, producing chemicals, the inflammatory explosion, as I mentioned. So let's say this is setting the fire. Okay? Now, naturally, because every time you get infected, you mount an inflammatory response. That's why you feel sick, actually, of course. But you know, after a day or two, that goes away. So you turn it off. Okay? So there is a way of turning off inflammation. Actually, until about 15 years ago, no one ever really thought about this. And all of the focus of the pharmaceutical companies and so on was on blocking the initiation. Non-steroidal anti-inflammatory drugs, leukotriene receptor antagonists, all of those things. But then people suddenly realized, well, actually, maybe this turning off phase is pretty important. And maybe in inflammatory conditions, one of the problems is not the initiation, but it's a failure to resolve, a failure to turn inflammation off. So in other words, a failure of the fire brigade to either come or to function very well. So the fire keeps going. Okay? So people have been very interested in the resolution of inflammation. And it turns out that there are chemicals that turn off inflammation that are made uniquely from both EPA and DHA. These are called pro-resolving mediators. They've been given the name resolvins, protectins, and neuroprotectins and maressins. So The pathways are probably not of interest to you. They're very complicated. People haven't worked them out in full. But there are pathways by which EPA can be converted to resolvins. Interestingly, in the presence of aspirin, some resolvins with slightly different structures are formed that are also biologically active in turning inflammation off. And DHA is even more complicated, so there are 6 different resolvins. There are some aspirin-triggered resolvins, so these are produced in the presence of aspirin. There's some protectins, and there are also now several of these moresins. So these have mainly been studied by a group in Boston, in the US. I just show you data, I think, from one animal study, just to show you the sort of thing that these resolvins can do. So this is a mouse study of colitis. So in this study, colitis is induced by exposing the animals to a chemical that causes the colon, it irritates the colon, it causes it to shorten and thicken, as you see here. The animals lose body weight, a lot of them die, and as I said, the colon changes its length and morphology. If resolvin E1, so this is a chemical produced from eicosapentaenoic acid, is administered, the body weight reduction isn't so much, the survival is increased, and the change in colon length and morphology isn't seen. This is a readout of the movement of white cells into the gut mucosa, so into the gut wall. This is massively upregulated by this chemical. That's what's actually driving the whole thing. If resolvin E1 is administered, that doesn't happen. And then these are all inflammatory chemicals. A lot of them are increased with the agent that causes the disease. And if they're increased by that agent, then in general, resolvin E1 prevents that from happening. So these are pretty potent chemicals. So with regard to these, what are called lipid mediators, so these are chemicals produced from fatty acids. The omega-6 fatty acid, arachidonic acid, gives rise to chemicals with high pro-inflammatory potential. This is prevented by EPA. EPA gives rise to chemicals of low pro-inflammatory potential, and both EPA and DHA give rise to inflammation-resolving chemicals. This is one of these cell signaling pathways that I talked about before. So this is the cell membrane here with the membrane phospholipids, and then these are receptors. And these are receptors for different things that can trigger inflammation. So this is a chemical, it's a protein called tumor necrosis factor. This is part of bacterium, this is bacterial lipopolysaccharide. This is another inflammatory chemical, interleukin-1, this is UV irradiation. So there are receptors for all of these things. Then there's a signaling process that I talked about before, lots of things happening in the cytosol of the cell. In the end, what happens is the nucleus becomes activated to express genes involved in inflammation, and no proteins encoded by those genes are expressed. This signal to the nucleus is caused by what's called a transcription factor. This is a protein which moves from the cytosol into the nucleus, binds to the DNA, and gets the gene expression going. In inflammation, one of the main transcription factors is this one. It's called nuclear factor kappa B. I put this in because I had the slide and it was a bit different from some of my other slides. This sort of process is very important in arthritis, inflammatory bowel disease, asthma, all of those things, but also in people bearing advanced tumours. So what happens is actually both the immune system, the inflammatory arm of the immune system, and the tumour are producing these very strong pro-inflammatory proteins called cytokines. Those proteins cause anorexia and cause loss of muscle protein, body wasting. Now we've got muscle tissue loss. This obviously reduces quality of life and increases morbidity and maybe even mortality. So these chemicals are important in classic inflammatory conditions and in people bearing tumours. So this is an experiment. I've got to explain what's going on here. These things are readouts of different inflammatory chemicals: cyclooxygenase 2, nitric oxide synthase, interleukin-1. Now this is a readout where the cells are not stimulated in culture. and then this is where they are stimulated in culture, and you see this big difference in upregulation of these inflammatory genes. And then this is if increasing amounts of the omega-3 fatty acid DHA are included in those cell cultures. So you see this marked reduction and almost abolition in some cases of the inflammatory readout. So in culture you can make cells have an inflammatory explosion, and you can prevent that by exposing them to omega-3 fatty acids. And in this study, to cut to the chase, the effect of both DHA and EPA was shown to involve an interference in this process here, okay? And people are very interested about that. That process is triggered by bacterial lipopolysaccharide and by some inflammatory cytokines, but interestingly it's also triggered by some saturated fatty acids. And there are experiments that are showing that omega-3 fatty acids don't just interfere with the signaling caused by the classic inflammatory stimuli, but also by saturated fatty acids. So that actually also means there's some interrelationship between saturated fat and omega-3 fatty acids in terms of driving inflammatory processes. Just if you're interested, about 3.5 years ago, actually it's 4 years ago now, some Americans showed there's a receptor on the surface of inflammatory cells for omega-3 fatty acids. So just like hormones have receptors and fat-soluble vitamins have receptors, people have discovered omega-3 fatty acid receptors now. And these receptors act in an anti-inflammatory way, although more work is needed in this area. So what I've been telling you so far is that if you give people more EPA and DHA, you change their cell membranes, and I've focused on membranes involved in inflammation, but I mentioned platelets and other things in passing. This changes aspects of membrane structure and function, and I guess structure-function relationships, changes the substrates for eicosanoids like prostaglandins and leukotrienes, and for resolving mediators like resolvins and protectins. And there are also these receptors that I mentioned. So all of this is about signals. Signals that cells are sending outside, and signals that cells are sending from their membrane to their nucleus. So we're really changing the way the cells are responding. Let's skip this. So what this means is omega-3 fatty acids could be useful in any condition that involves inflammation. Okay? And actually, they've been tested in every condition here, some to a greater extent than others. So I'm probably going to talk about a couple of these now as an example. So the first one is rheumatoid arthritis. So if you're interested, this is a review that Liz Miles and I published in British Journal of Nutrition in 2012 on EPA and DHA in the context of rheumatoid arthritis. So there's a genetic susceptibility to arthritis, of course, and that susceptibility is something to do with immunity and driving inflammation. It's an autoimmune reaction. If you take fluid from the joints of people with arthritis, that fluid contains high amounts of the sort of chemicals that I've just been talking about, tumor necrosis factor, interleukin-1, and what have you. And if you take the cells from the synovium and put them in a test tube, they just chug out these chemicals. So also the enzymes that metabolize arachidonic acid are upregulated in the synovium and also in animal models in the joints. And of course people use non-steroidal anti-inflammatories to treat pain and stiffness and arthritis. And all of that is driven by arachidonic acid derived mediators. And those chemicals made from arachidonic acid are found in the synovial fluid. So people, actually the rheumatologists were almost the first people outside of the cardiovascular arena to recognize that omega-3 fatty acids might be important to them. So people started studying omega-3s in arthritis in the 1980s. It works, I put it in inverted commas, in other words it can both prevent and treat animal models of arthritis. If you give sufficient omega-3 fatty acids to patients with arthritis, you can show changes in the sort of chemicals that I've been talking about. There have been 20 plus proper randomized controlled trials of fish oil and arthritis. There have been lots of other sorts of studies, open label studies, all sorts of things. But there have been over 20 randomized controlled trials. The first was reported in 1985. The most recent, when I made this slide, was 2009. Some of these have used what I would call a pretty low dose. Some of them have used a very high dose, more than 7 grams of EPA plus DHA, not a fish oil, of EPA plus DHA. Because remember, most fish oils are only a third, maybe 40%. If you get a concentrate, maybe 50% of EPA and DHA. So this is an awful lot of fish oil. Could be as much as 20 grams of fish oil. That's a lot. Durations from 1 month to a year. There are actually not many dose-finding studies in arthritis, but they look at different times and they report all sorts of things. So out of 23 studies that we talked about in this review, 17 of them reported improvement in at least 1 clinical outcome, 14 in at least 2 clinical outcomes, and 6 in at least 4 clinical outcomes. And these were things like reduced number of swollen and tender joints, decreased pain, increased grip strength, decreased duration of morning stiffness. And the really interesting thing is all studies that monitored NSAID use reported a significant reduction. So there's an NSAID sparing effect. So if you think about what I've been saying, a patient would take non-steroidals because they felt pain. Okay? If omega-3 fatty acids are changing the balance of chemicals so there's less pain, a patient won't need to use NSAIDs. So that's the explanation for what's been observed in these studies. This is detail from the review. You can look at it if you like. There have actually been some meta-analyses of omega-3s in fish oil that report significant benefit on quite a number of those outcomes when the trials are aggregated. So the evidence, I think, in arthritis is pretty good. It's actually the inflammatory condition with the best evidence, I think. The second area I want to talk about, just out of interest, is this area of advanced cardiovascular events, because that's a little bit different. This is a cross-section through a blood vessel wall. So the blood is flowing this way. Okay? This is the endothelial layer that I mentioned before. And this is a person with an advanced atherosclerotic plaque. In other words, they've got a big fatty build-up within the vessel wall. Okay? So there's all of this lipid and necrotic tissue. Smooth muscle cells have actually moved from where they should be over the top of this. They've produced these proteins, things like collagen and fibrin. This is called the fibrous cap. And this is probably an attempt to just try and keep all of this contained. It's like throwing a net over it and saying, you know, please stay there. Like you see on the side of motorways up those walls where they're trying to hold everything back. So what's happening here also is you see these cells in the vessel wall. These are inflammatory cells. And it's recognized that actually all plaques have a lot of inflammatory cells associated with them. And these cells, they're all communicating with one another. They're producing the chemicals that I've been talking about. And some of them that are produced are these things called matrix metalloproteinases. These are proteins that degrade other proteins. So these MMPs actually degrade the fibrous cap and make it thin. So it's a bit like getting some bolt cutters on those nets on the side of the motorway and making them less able to hold back what's there. Okay. So what happens then actually in these thin capped vulnerable plaques is you can get a rupture at the edge. So there's a break. What happens then is you get platelet aggregation and thrombus, and the person has an MI or a stroke or something like that. So inflammation seems to be pretty important. We saw the front cover of Scientific American. Inflammation's linked to heart attacks. And this is exactly what it's about. So about 10 years ago, actually, let me just show you this first. So I'm going away from inflammation now to show you this trial from Italian researchers published in The Lancet in 1999. So they recruited 11,000 patients who had had a myocardial infarction but survived. They'd had that within the last 3 months. And they were assigned to one of 4 treatment groups. About half of the people in the study received omega-3 fatty acids in a particular preparation, and a quarter of them received placebo. There was another group that was about vitamin E, but that didn't have any effect. These people were followed for 3.5 years, and these are these are the differences between the omega-3 group and the control group. So these are the percentage reductions in mortality outcomes, okay? So there was a 20% reduction in fatal events with omega-3s compared with placebo, okay? 30% reduction in cardiovascular death, 35% in coronary death, and 45% reduction in sudden death. Other deaths, cancer, road traffic accidents, all of those things were no different between the groups. Okay? All of these were highly statistically significant because the study is so large. Okay? 5,000 people in one group, 2,500 in the other. This is a massive study. So even a 20% reduction can be p less than 0.001. Okay? Because the study is so big. So this is a study which showed that giving people who are at risk omega-3 fatty acids reduces the risk. No one could explain that. They thought it might be something to do with cardiac arrhythmias, all sorts of other things were thrown up. But we thought we could explain it because we said, what happens if omega-3s get into the vessel wall and just calm down the inflammation? So that was our hypothesis, that omega-3 fatty acids would stabilize plaques, making them less likely to rupture. So we had to study this somehow. So essentially, we needed to get hold of plaques from humans to whom we gave omega-3 fatty acids. Okay. Now, fortunately for us, there's a surgical procedure called carotid endarterectomy. So this is surgical removal of advanced plaques from the carotid artery. Okay. So this is a cartoon of what happens. So you clamp, well, you expose the carotid artery, you clamp it off, you slice it open, and you pull out the plaque. Okay, this is it happening in real life. Okay, this is done daily in every hospital in the UK. Okay, because these people are at great risk of stroke. Because you'll see the size of the plaque. They have 80% plus occlusion of the carotid artery. Okay. So this is an essential operation which is carried out probably in the tens of thousands in the UK. I don't know how many are done a year. I haven't done the calculation. So we did a study on these people. The other thing that is very fortunate in the UK system, of course, is, when Mr. Smith knows that he needs this life-saving operation, he doesn't have it tomorrow or the day after tomorrow. He has to go away and wait many months. Okay? So then you can do a trial on Mr. Smith and all of the people like him. Okay? So we did a randomized controlled trial in 180 patients awaiting carotid endarterectomy. We did nothing else to interfere with their clinical practice except we gave them vegetable oil, another vegetable oil or fish oil, from when they knew they were going to have the operation until they did have the operation, which on average was 7 weeks later. So what we discovered, this is old now, but it's still my favourite study that we've done of all time. So what we showed for the first time ever was that if you give people omega-3 fatty acids, EPA and DHA, they end up with more EPA and DHA in their atherosclerotic plaques taken at surgery. So even though some of these people were in their 80s, their plaques have been building up for 79 years. Within an average of 6 weeks, you can get omega-3s into the plaques. So the plaque's actually quite dynamic. Actually, cells are moving in and out of there. Lipid is moving in and out of there. They had fewer inflammatory cells within those plaques. And we had the cardiovascular pathologist, whose job it was to do this every day of every week, so he routinely, his job was to look at the plaques from every surgical procedure and grade those plaques. So there's a particular grading system that's used. It's an American Heart Association system, and he showed, or his numbers told us that the plaques coming from people treated with omega-3s were more likely to be stable than unstable compared with the control group. So this was pretty remarkable. Association of omega-3s with stability of atherosclerotic plaques, a randomized controlled trial. This was so interesting to the pharmaceutical industry that a company approached us with a checkbook and said, will you do that study all over again with our product? Now, we said yes, of course, but we wanted to do some science. So we repeated the study with an identical design, except we only had two groups this time, vegetable oil and control group, and the science we wanted to do was look at inflammation in the plaques, because we didn't have any material left from our first study. So essentially these are all inflammatory chemicals, including some of these matrix metalloproteinases that degrade the cap and make it thin and vulnerable to rupture. The control group is in grey, the omega-3 group is in black, and you'll see that whatever's measured is always lower after omega-3s than control. So remember, these people had built up their plaques over decades, and in a few weeks of treatment we could reduce the inflammation. So what we think is increasing availability leads to incorporation of those fatty acids in the plaque. They do all the sorts of things that I've been talking about already. They basically turn off the inflammation, reduce the inflammatory burden. They come along and help put the fire out, or at least dampen the fire a little bit. So now you've got less inflammation, increased stability. People don't have events, or they have weaker events. They don't die. So that was our explanation for the Italian study. The Lancet bought that idea, which is why we could publish our paper in the Lancet in 2003. This is my last slide. I told you inflammation underlies many common conditions and diseases, and there are characteristic markers that can be measured. Human inflammatory cells typically contain a lot of this omega-6 fatty acid, arachidonic acid. That's a precursor of known inflammatory chemicals. What I've called marine omega-3s, EPA and DHA, easily get into these cells. They do all sorts of things that I've bored you with about membranes and chemical mediators and transcription factors and signaling and stuff. So what that means is these fatty acids, when you've got more of them in your cells, you can counter the effects of classic inflammatory stimuli. There's good evidence of that. They have a role in controlling inflammation in people with frank inflammatory conditions. I talked a bit about arthritis. And probably they impede the development of what's called chronic low-grade inflammation. So that's the inflammation involved in the vascular wall, in obesity, and non-alcoholic fatty liver disease. You wouldn't call those inflammatory conditions. And you don't treat those people specifically with anti-inflammatory drugs to control inflammation. But there is this ongoing low-grade inflammation. By low-grade, I mean the chemical markers you measure in the bloodstream, of course, are not as high as they are in someone with frank arthritis, for example. So it's a lower level. But I think there's a role for the fatty acids in both probably in prevention, but in treating those sorts of conditions. That's it. Thanks so much.